Now I'm recording. Try to share screen again. So hopefully it's still just one big PowerPoint slide, mostly blue. Well, we're all staying dry, hopefully. If most of us, I'm guessing, might have stayed at home. But so today, I wanted to talk a little bit more about how to survive tough times, and then get into a little bit of physiology and think about how I want to give you enough knowledge on physiology so that you can think about it from an ecological perspective of, given a certain habitat, what kinds of microbes might be there and what kinds of strategies would they have to stay alive, get access to nutrients, reproduce. So I think we left off here looking at the idea of, how to beat tough times. We know that you can have kind of cannibalistic effects where microbes can cannibalize each other. That endospores can be a great strategy, a very long-term strategy if needed. There's other types of spores that aren't quite as resistant as endospores, but still a decent way to go dormant. When you think about microbes that are going in, starting to experience a tough time, one of the other things that they can do is just slow themselves down. And so if they can get more into a holding pattern or a resting state phase where they're ramping down their physiology and their activities in their cell so that the metabolic demand is lowered. And one thing that they can do to achieve this is they can actually shrink themselves, which seems weird, right? Because as humans, we tend to grow to our mature height and well, not necessarily weight, but mature height at, teenage years. And that's pretty much it. And then I guess when you get really old, you start to shrink, I'm told, but in height, a few inches here and there. But microbes have this ability throughout their whole life to fluctuate the size of their cell. And so experimentally in the lab, you can see this happening where they put the microbes under a starvation condition and then they actually measure the cell size, and it can be appreciably smaller when you look at the cells under starvation. So it's kind of cool how they can just, go really small, and that really does help them slow everything down inside themselves. So their metabolic rate slows down, their growth rate really slows down. They really don't bother with reproduction very much at all. And so respiration is talking about their physiology. They just kind of ramp things down and try to kind of just go into a sleepy phase, so to speak. Often when you look inside microbes, you can, if they have gone through a feast condition where they've had ample access to food, one of the things that microbes can do is store up some food for thinking ahead, so to speak, where they can have a reserve inside their cell, especially of phosphorus and carbon. And so those two in particular, you tend to see certain polymers and complex molecules that can get kind of tucked away inside their cell when they're undergoing really flush feast conditions. And then when they enter into the starvation state, they can start to tap into that and hopefully make it through the tough time. I'm not going to go into details, but they have studied this idea of protein turnover. So they'll see some proteins get degraded, and probably some of the amino acids might get then put into other physiological processes, and some of the nutrients that make up the proteins. You get complete protein breakdown so that they can kind of cannibalize their own proteins as a nutrient source, if you want to think of it that way. And then typically they do tend to have the genes necessary to take advantage of a flush of nutrients up and running, or at least very quickly to be able to turn it on. And again, this is done through experimental lab-based studies where they can show that once you have them into like a full starvation state, then if you introduce some nice nutrients, they seem to be able to very quickly take advantage of that. So they have the necessary genes to be assessing their environment and go, oh, we just got a flush of glucose. Great, I can turn on this pathway and immediately start using it. And so that's another thing that they've noticed when they play with this in the lab. And then some microbes they'll do enough differentiation of their cell, so they'll miniaturize, but they can also put appendages on themselves. So prostheque is just a fancy word. Another word for that is a stalk. We've talked about bacteria that can have a stalk for attachment. And so sometimes when they go into this, oh boy, they're assessing their environment and they're thinking, well, you know what I mean, they're not thinking, but they're assessing their environment and they realize, oh, we're running low on, nutrients, water. Sometimes they'll try to stay put in with the idea that what the hypothesis is that the stalked bacteria might get localized food, better access to localized food, at least for a little while. They won't get swept away, for example, in the stream. Because if you think about a stream, for example, and think about the size of a microbe in comparison, it would often be a better decision, so to speak, to stick onto a rock than to be swept away into the middle of a stream, because typically nutrients, you get far more nutrients on a rock surface that the microbes can access compared to the middle of a stream, for example. Much more diluted out in a fast-running stream. So depending on the habitat, you might see some bacteria make the choice to go ahead and make a stalk or a prostheque to attach. And so this is not an exhaustive list, but these are some of the things that they've noticed when they play around in the lab with starving the poor microbes and watching what happens. Then also, depending on the situation they set up in the lab, you can see that the microbes might shift their morphology quite extensively into a different stage of their life cycle. And it allows them to disperse away from an area that is, there's no nutrients here. So they may shift their morphology in such a way to be able to pretty efficiently move out of that zone of, there's no nutrients here, we need to move out. So I want to give one example of that. And this is the idea of swimming and swarming processes that go on. And this is most heavily studied in Proteus species. Proteus mirabilis is a common microbe that we work with in like Gen Micro, for example. And if you grow it on an agar surface, it tends to form this very distinct pattern, colony growth pattern. And I'll show you that in a minute. But why it's making that pattern on the agar is this idea of, normally the Proteus looks like what you see on the left image here, where it's a rod-shaped cell with some flagella around it. And then what happens is it just says here, environmental signal. It's usually it can often be a starvation situation where they recognize there's no nutrients right around them. So then they'll switch into this shift in their morphology. So the cells greatly elongate, and look at how many more flagella get made, which is impressive because the flagellar structure is quite complex. And so they make a lot more flagella. So there must be a benefit to this, because that's definitely a cost, right? Making all that flagella. So they elongate and make a lot more flagella, and then they're called a swarmer cell. And what happens is you get a bunch of these Proteus cells doing this all in one area, and it causes them to then like come together as a team. And so there'll be this bunch of them all with these elongated cell structures and flagella, and they'll start to move across the surface en masse, like as a team. They'll move across the agar surface, for example. And the hypothesis is that they're doing that to get to a new habitat where hopefully they can find more food. Plus, if they team up, it seems that they can release their enzymes as a team and more aggressively attack any nutrient source, whether it be a very large molecule or maybe a small organism that they come across. And so if they're this big mass, they can take on even small eukaryotic single-celled organisms and kill them and use them for nutrients. And so it's an interesting process that they'll kick into when you place them under starvation conditions. So Here they're just showing some of the characteristics of the swimmer versus the swarmer cell type. Let me go into the next slide here. So when you do this in the lab, they often work on agar plates, and they will create an agar recipe that's very devoid of nutrients. So very quickly, what they tend to do is just place like a loop full of the Proteus right in the middle of the plate. And then the recipe is so devoid of nutrients that pretty quickly the Proteus that is there will run out of nutrients, and so they have to move out from the spot where they were originally inoculated. And so that they do need a solid surface. If they don't have a solid surface, they can't swarm. It won't work. We've never seen them do it in a liquid. It has to be on a solid surface. So if they're on that solid surface, if you push them into a starvation state, that's when they will elongate their cells. Lots more flagella get put into their cell design, and then they start grouping together and swarm over the solid surface. And they've experimentally done this where you can't, like they've teased apart and separated out the swarming cells from each other and like plopped, you have to have really special, I think they're called laser tweezers to do this. Kind of sounds like a fun gadget to play with in the lab, but you have to, when they put just one swarmer cell separate from the bunch, that swarmer cell just stopped behaving as a swarmer. So they, again, suspect that there's quorum sensing going on, where these swarming cells are assessing how many of me are around me, and when there's a threshold level of the cells all together, then they can start to move as a team across the surface. And so it's this pattern of, you can see it toggle back and forth between their swarming phase of their life cycle, and then, for example, experimentally, all of a sudden they'll give the swarming cells a flush of nutrients, and then the swarmer cells will say, oh, you know, I have a bunch of nutrients, I can go back to my swimming phase of my life cycle, where they can shrink down, abandon some of those flagella, and be the more typical cell that you think of when you think of Proteus. And so it can go back and forth like that. At least experimentally, they can play with that in the lab, and they can show that the microbes can go back and forth. And it's, what I put here, I don't really care that you know these differentiation consolidation terms. If you're interested, you can read more about it, but it's the big picture idea I want you to know is that you understand the difference between swimming and swarming, how the microbes, what they have to do to enter into the swarming phase, and that they can go back and forth depending on the conditions they're exposed to. And again, here they're just trying to show that this is cyclic, that you can get the, you know, switching back, swimmer, swarmer, swimmer, swarmer. It just depends on the habitat and what they're exposed to, how they will move through their different phases of their life cycle. So kind of a fun thing to play with in the lab. This is what an agar plate that was spot inoculated with Proteus looks like. So they put Proteus right in the middle. They just took a loopful of broth of Proteus, and then they put it right in the middle and then incubated the plate. And so you can see this bullseye pattern, I guess is what you would call it. And so what's happening is they are swarming, and then you see this kind of break. Here's where they went back to swimming. So in this area of the agar, they got access to some nutrients. So they went back into the swimming phase. Then they ran out of nutrients. So then they got, you know, they gathered together as a team and started swarming. So then this next swath of that light tan turbid look, that's the swarming phase. And then you see another demarcation here. That's the swimming phase. All right, so swimming, swarming, swimming, swarming across the whole plate. And they can cover the whole plate if you let this plate incubate long enough. Now I have a video. So here, this is another, it doesn't always look like what I just showed you. Sometimes the swimming, swarming patterns can look quite different. Proteus is the classic example, but here on the left, this is Pseudomonas aeruginosa, which is a very interesting bacteria that can do all kinds of weird things, some good, some bad, and at least from a human perspective, they can cause disease. And actually, Pseudomonas is amazing. They can cause diseases in humans, and other animals, and plants. and insects, I believe too. So they are they have a very broad host range, and they have a lot of tools in their toolkit to cause disease in different hosts. It's really quite amazing. This one on the right, this is a rhizobium species that can do nitrogen fixation with plants. And most rhizobium are motile because they have to find the plant roots when they're hanging out in the soil. They have to, if they want to do this symbiotic effort with the plant, they have to be able to move toward the plant. So, and again, you can see it's a little bit different pattern, but you can clearly see that they're motile. Both of these were inoculated in the center of the plate and then allowed to incubate. And I think I have a video of Pseudomonas doing this. Let me see if I can actually get it for you to see it. I'm going to go into Google, YouTube. Can you see a YouTube right now? Do you see that we're in YouTube?
Yeah.
Let me see if I can get this to work. So on the left of this plate, you're going to see the Pseudomonas growing out. On the right of the plate is a mutant that was inoculated in this half of the center. That was deliberately, they created a mutant in the lab that cannot swarm. And look at the difference. It's just a short little clip here. So hopefully you're seeing the growth now, and they're starting, the left side starting to move out, swimming and swarming. But the one on the right is staying put. So pretty, oh, really short. Let me do it one more time. So pretty cool. Obviously they sped this up dramatically, but it's pretty fun to watch. So there's many, there's many bacteria that can do this. Let me go back. I'm going to stop. Oh, wait, let me pause this so it doesn't kick into some other video and start annoying us in the background. All right, let me go back to the slide. So now we're back to my PowerPoint slide. Hopefully we're seeing the two Petri plates. And so when they noticed this in Proteus, that was probably the first group of microbes, the first genus that they noticed this pattern. And then they, obviously here with Pseudomonas and Rhizobium, but you don't have to write this down, this list, it's available, so, and I'm not going to be asking you to memorize this list. I'm putting this here. The big picture idea for you to grasp is that there's been studies done on many bacteria that are often quite distantly related from each other, and they can do this. They can do this swarming behavior. And so again, when you see that it's kind of a universal or a very common strategy, then it gets you thinking about, okay, from an evolutionary perspective, this is probably something that definitely, the benefit outweighs the cost under many conditions that the microbes are exposed to. And so, with that, there's some advantage to it that many microbes that are not related to each other have evolved this mechanism. And again, it makes sense if you're this tiny little microbe and you're plopped down into a habitat that becomes depleted of nutrients, if you can't move and get out of there, your other option is to somehow go dormant or die, so it's kind of a pretty dire situation that many microbes can be put into throughout their whole life. I mean, especially when you think about the soil environment or, lakes and streams, you can appreciate how a tiny little microbe might not be in the best habitat and needs to move away and get into a new environment. So they do think that it's more efficient when they can team up like this and move en masse, and it also does allow for, some probably will die in the process, but at least some will survive to pass on their genes to the next generation. So it seems to be a good strategy that many microbes have evolved this behavior. So this table is looking at the idea of, thinking about, well, what does it take for a microbe to, we say doubling time, and that means how long does it take a microbe to double its population? So when you work with E. coli, for example, this first row here, if you work with E. coli in the lab, E. coli is probably the most heavily studied microbe out there. I remember for my PhD work, you have to go through qualifying exams where they make you do a written exam, and then if you pass that, you go on up to the oral exam, and you have to pass both phases in order to continue on into the PhD program. And when I went to my committee members and said, what do you want me to focus on for studying? One of the committee members gave me this big, thick volume, and it was all about E. coli. It was all about, the physiology of E. coli, anything you wanted to know about E. coli. And he's like, here, read this. And it's like, oh, great. Yeah, no problem. I'll read the whole thing. So it was, they know a lot, and a lot has been published, books and thousands of published papers. Anyway, so we know how to grow E. coli pretty easily in the lab where we can optimize its growth rate. So 20 minutes, that's all it takes for E. coli to double its population. But if you think about a more natural habitat, like we have billions of E. coli in our intestinal tract, normally, not the ones that cause disease, and that can take 12 hours because, of course, it's not optimized. It depends on what you eat. How are you feeding your microbes? Remember, you need to take care of your microbes. We'll talk more about that later in the semester, but what you eat can profoundly influence the microbes that hang out with you, both good and bad. And so we'll talk about that. And then some other habitats, the mouse, inside the mouse intestine, 10 to 24 hours for salmonella. And then just looking at some more natural environmental systems, how long. And so you can see it can vary. It just depends on the habitat and their particular access to the nutrients. Soil, you can see it's pretty tough life out there. 100 days it can take for some microbes to double their population. And then down here it's looking at dormancy, duration of dormancy or survival. And I mean, look at this. This is impressive. Clostridium, which is an endospore former, 34 years it survived in apparently that was just in a laboratory setting. Lake Vostok, that's over in Russia, I believe. 1.4 times 10 to the 5, so that's, many zeros after the 1.4. It survived, and the gut of an extinct bee trapped in amber, they isolated some heterotrophs. That just means there were heterotrophs. We have to take in carbon from something that was once living and use that in order to get our carbon needs met. And then bacillus is a endospore forming microbe. And look at that, 25 to 40 times 10 to the 6 years it survived. So that's how old the amber was. They picked out that bacillus from the amber and worked with it in the lab and got it to grow. So they're saying that it survived going into an endospore state for that long. And look at this last one, this endospore, when they found it in some fossilized rock material, and 250 times 10 to the 6 years. That's just incredible to think about that it could stay dormant, and then when they plucked it from these habitats and put it into the lab environment, they got it to grow. It's just amazing. That would be so cool to work with something that old. One time in my life, I'd like to get that experience. I just think it's so cool. And look at this. This is amazing. So this business was looking for unique yeast to brew beer, and so they did this too. They went into amber and isolated out yeast from amber, revived it in the lab, and then brewed beer from it. Now, I think that's pretty cool. I like that idea. I think that's an interesting twist. And of course, it's a good marketing tool, right? Especially for any of us biology geeks that get into that kind of stuff. So you can read this on your own time, but they said, toward the bottom here, it said, aside from that, it produces some secondary metabolites that add unique and interesting, pleasant organoleptic characteristics to the beer. I think organoleptic means, what your taste buds are tasting. I think it's a fancy word. So pretty cool, don't you think? Brewing beer with, tens of millions. I think it was 45 million year old. Maybe I can't remember where it is. It's in here somewhere. I think. So, pretty cool idea. Maybe we should tell the people at Black Rocks or the ORDOC, they should get that and start marketing it that way. Because from what I understand, a lot of biology professors go to Black Rocks on a regular basis. So we could probably market it to them, right? Okay, so that's a little bit on microbes surviving tough times and the amazing longevity of some microbes. But now I wanted to get a little bit more into some basic physiology of the microbes. Do you see one big slide again? Mostly blue?
Yes.
Okay, thank you. So what I'd like to do for this is try to get you thinking about what you might have already learned about basic physiology. And so a lot of students that are in my classes, they've had like animal phys or human phys at some point, or just through their own readings, they're a little bit up on what our bodies can do for physiology. So I like to kind of start there and get that part of your brain firing, and then I want to layer on this new information. By the way, when you're studying that, learning new material, layering it onto something you've already learned, building on that knowledge foundation is a really good strategy. And that's one of the, I'll tell you a secret, as you get older, you learn faster because you are building such a base of knowledge. So, and I have seen this even from freshmen that I advise, the freshman students here at Northern. And then when I meet with them, through their career at Northern, and then they're at their senior year in, like their last 2 semesters. And I often get into conversations with them where they can realize that it's easier for them to learn, into their senior year because they built such a base of knowledge that when the new information is coming in, they can process it better and get it into their long-term memory because they're making associations in their brain quicker, and they're linking the information. And then when you get into your real life beyond Northern, I hope you continue learning, and you will see again that it just comes easier and faster, and you can solidify the information more quickly. So that's one good thing about getting older, is that you learn faster, and it usually is less of a burden, or it's less of a, it's easier. It's easier. So anyways, so let's look at this. So we're thinking about biochemical reactions that are happening in the cell. Why do microbes do it? A huge reason is to get ATP, because that's the chemical currency that all living organisms use the most. And so we're going to focus on the so-called catabolic reactions. There's another category of reactions that we call anabolic reactions. Anabolic is to build. So if you think about taking anabolic steroids to build muscles when you're weightlifting, for example, some people will take anabolic steroids to build up muscle tissue. So you're building up, you're making bigger structures, polymers, polysaccharides out of simpler molecules. That's a different thing. Catabolic, the way I link this term and memorized it when I was an undergrad, is that at the time I was taking biochemistry, we had a family cat that had its claws, and one of its favorite things to do was to climb the curtains in the living room, which would drive my mom nuts. And then if we saw the cat high up on the curtains, we would yell at it, and then it would kind of like lose its grip a little bit and slide down, and it started shredding the curtains. And for some reason that stuck in my brain as I was learning about catabolic reactions, my cat shredding the curtains. So catabolic reactions shred molecules coming in, they break them down and push them into physiological pathways that it links it to excuse me, ATP production. And so catabolic is the breaking down of molecules, and through that process you get ATP production. So in our bodies, I shouldn't say that. In general, when you think about all living organisms, there's 3 big categories of how you can synthesize ATP. Oxidative phosphorylation, that's what we do in our mitochondria. So I'll talk a little bit about that. Substrate level phosphorylation is another way to make ATP, and we do that too when we do glycolysis, but there's lots of different substrate level phosphorylation processes in microbes. And then photophosphorylation, sadly we can't do that one because that's looking at photosynthesis, and unfortunately we can't go to the beach, lay out in the sun, and make food. It'd be awesome if we could do that, but we can't. So, but these are the three big categories, and I would like you to be able to, state these if I ask about them, because this is kind of like the big picture idea, like, okay, of all the organisms out there, how are we seeing ATP production? All right, so let's look at some of these now. So glycolysis is an example of substrate level phosphorylation. You've probably heard of the citric acid cycle or the Krebs cycle. Some call it the tricarboxylic acid cycle or TCA cycle. It's all the same thing. I tend to say citric acid cycle. And so that is another link in the chain to get to ATP production. And there is some ATP production in that cycle, but a modest amount. And then the electron transport chain, which is where you really see some really decent ATP production going on. So you've probably heard about this at some point, especially if you've taken a phys class. That should sound real familiar. And I talk about it quite a bit in Gen Micro. We get into the different ways microbes do their physiology. So the electron transport chain in our mitochondria, that's what they refer to as oxidative phosphorylation. And so we're going to look at some of these, and then we'll start looking at variations on that process. Let me just check here. All right, so what do we got here? Oh, yeah, so I think if you took Gen Micro with me, you certainly understand that microbes are amazing in their ability to run a variety of different physiological reactions. And one microbe can have a really wide repertoire of physiological reactions to put into play under different environmental conditions. And then when you look across all the microbes that have been studied, some of them are just like, it's almost like science fiction when you read about what they can do. It's crazy because it's so different from what we do. We're so limited compared to what microbes can do. We're kind of pretty simple organisms when it comes to that, even though we are an amazing organism, but our physiology is pretty simple. So anyways, let's look at this. This is a big picture idea where they've looked at glycolysis, the citric acid cycle, and the electron transport chain. So you're looking at a cell, some microbe that is running all of these. And so let's start over on the left. We see here the idea of glycolysis, and this is very abbreviated. Of course, you're not seeing the full pathway. But here they're saying that one of the main events in glycolysis is we normally think about starting with the glucose molecule. And when you put glucose into the glycolytic pathway, about halfway through it, you end up with two pyruvate molecules. And that's a key part of the pathway because once pyruvate gets made, depending on the habitat of the microbes, pyruvate can get shunted into other pathways. So one of the big decisions the microbe has to make is if oxygen is available or not. And of course, you have to be the type of microbe that can use oxygen for ATP production. Not all microbes can do this. But let's think about E. coli. E. coli can do this. If they have access to oxygen, they will take the pyruvate that they have made through glycolysis and they'll push it into the citric acid cycle, the TCA cycle, the citric acid cycle. So that would be the next step. And in the citric acid cycle, through that process, you do get ATP a little bit, just like in glycolysis. You get a little bit of ATP. You get a little bit more in the citric acid cycle, and you can see that CO2 is getting respired off, all right? So they're going through the respiration idea of giving off CO2 in this process. And then you can see that they will feed some of the electrons, some of the molecules that are created through these pathways, some of those will get fed into the electron transport chain, because now with the electron transport system, that whole thing is transferring electrons from donor to acceptor down the chain. And as that is happening, it sets up a situation where you can get lots of ATP. So here you see 34, in this particular case, 34 ATP molecules made through this process of the electron transport system. So you can see that of these processes going on in the cell, it's definitely the electron transport chain system that's the big payoff. But you have to have oxygen in this particular case for this microbe, because you see here it says 6 oxygen. All right, so this microbe is using oxygen. And when you do that, you get a lot of ATP. Now, can microbes do some other kind of electron transport chain and maybe not use oxygen, have some other molecule? Absolutely, they can. But it's not as good as oxygen. As far as we know, oxygen is the best electron acceptor. It loves, chemically speaking, it loves to take electrons. And that sets up a situation where you get maximum free energy to get maximum ATP production. So oxygen, if you are a microbe, your smartest move would be I'm going to use oxygen and run an electron transport chain system to get maximum ATP. But E. coli, sometimes you can plop E. coli into a habitat where there isn't oxygen. But E. coli is so versatile. E. coli can go, oh, okay, well, now you put me into an environment with no oxygen. Well, that's okay. I'll just switch my physiology to some other pathway, and I'll still get enough ATP to survive through this. But again, if you present oxygen to E. coli, it would definitely switch over to what we see on the screen here, because that's where you get really good ATP production. So we're talking about ATP production, so we need to think about this very, very special ATPase enzyme that's embedded in the cell membrane of the bacteria. And so here you can see it's very complex, many subunits that make up the entire enzyme. And it's actually a little, they call it a molecular motor, because it actually, there's a torque force that gets generated as these protons go through the ATPase enzyme. It actually creates a torque force here where these subunits will shift and move. And as they're doing that, it allows for ADP and inorganic phosphate to react with each other. And so if you got everything going just right, these molecules will react with the flow of the protons through the enzyme, creating a torque force that allows these subunits to move, and that allows these molecules to react, and then you get your ATP production. I mean, sometimes, over the long weekend, if you're over 21, have a beer and think about this. I mean, this is going on in your mitochondria right now, and it's pretty amazing when you think about all the stuff going on at the cellular level. You got this little molecular motor working hard for you, getting your ATP needs. It's pretty amazing. So, okay, so let me go back a little bit. So this structure here is the ATPase we were just looking at. So they're trying to show the flow of protons through here. So we need to get some kind of proton gradient established, all right? The artist, and I wish they would have drawn a little bit more protons building up on the outside of the cell here, so they could more clearly demonstrate that there's a proton gradient developing across this membrane. So we'll come back to that idea. Any questions so far?
Yeah, Dr. Mackey, what did you say the slide that was showing the torque mechanism with the protons?
Mm-hmm.
What is that called, that unit?
You mean this area here, or just the whole thing?
Just the conglomeration of proteins and its function.
So it's called the ATPase. So you spell it out, ATP, and then in small letters, ASE on the end. So ATPase. And some people call it the ATP synthetase, if you want to get fancy. But most people just call it the ATPase enzyme. And very, complicated design, a lot of subunits.
Okay, thank you.
Mm-hmm. Any other questions? Okay. All right, so we can do some level of fermentation in our cells. Glycolysis is something that we can do. microbes as well. And so if you put a microbe into a habitat without oxygen, so anoxic means AN in science, anytime you see AN on the front of a word, anoxic, that means without oxygen. So we're talking about microbes that are anaerobic. And so if you're anaerobic, if you're an anaerobic microbe, that means you, and if you're an obligate anaerobe, that means you have to be in an environment with no oxygen. There are some anaerobes that are facultative. They can survive without oxygen and switch to fermentation. But then if you give them oxygen, they'll drop the fermentation and switch over to the electron transport chain process. And so with fermentation, when you look at, why do we do it? Why do microbes do it? It's to get ATP. That's the goal. So not a lot, but it's enough to keep you going. And when we look at the pathway and how this is done, it's done through the process of substrate level phosphorylation. So substrate level, that means when you look at a physiological pathway, you have the substrates that come into the pathway, and then you have, intermediary products as you work through the pathway, and then you have a final product. So you have a substrate going to products. So substrate level, and I'll show you a picture to try to drive this home. You have a phosphate group that's being transferred off of a substrate through this fermentation and being given to ADP in order to make ATP. So when you have these molecules in the glycolytic pathway giving off a phosphate, it's enough free energy when that bond gets broken and a phosphate gets given off to ADP. It's enough to cause for this reaction to get the ATP to be synthesized. So I'll show you a picture too, all right? And so when you have that phosphate coming off the substrate, getting cleaved off, that bond gets broken, that's a little boost of free energy to allow for ADP and the phosphate to react, and then you get ATP. So this is substrate level phosphorylation in a very generic way, the way it's written. So it's just, they're just saying, here's your substrate A coming into the pathway. Here are your intermediate substrates, so to speak. Here's your final product. All right, so substrate level, what they're saying is, so here you see a phosphate going on to this intermediate substrate called B, and you see the little squiggle. So when you have a squiggle bond like that, when they write it like that, that means that this has a decent amount of free energy. When that bond gets cleaved, it'll be a decent amount of free energy given off. And so it looks like the phosphate got transferred to C, whatever that is, and then here's the goal where you have ADP hanging out by C, and when that bond gets cleaved off and given to ADP, you get the reaction of C going to D, but the key thing is in the process you have the ADP and the phosphate reacting to get you ATP. So substrate level, when they say phosphorylation, they mean that ADP is getting phosphorylated. It's getting one more phosphate group, so it can be an ATP. T is triphosphate. So it went from adenosine diphosphate to adenosine triphosphate. So let's put that more into context of the actual glycolytic reaction. So this is glycolysis, and those of you who have had Gen Micro, hopefully this looks familiar, and it's don't let it cause nightmares, all right? So we're not going to get into the details. I will not ask you to memorize this. If you're taking biochemistry, yes, that's appropriate, but not for this class. So the big picture idea here you see glucose coming in to the pathway, and the yellow part of this pathway is what they call stage 1, where you actually have to expend a little bit of ATP to get this final fructose 1,6-phosphate in that version, that part of the pathway. Now they often refer to this as kind of like you're priming the pump. You're kind of getting things set up so that when you move into stage 2, you get some nice ATP production now. And so you do see that there's phosphate groups on this fructose molecule. There's 2 phosphate groups. And then you see it entering into stage 2. So now you see substrate level phosphorylation happening right here. All right, so you have 1,3-bis-phosphoglycerate, 2 phosphates here. And you have the ADP molecule. And so as you go into this phase of the pathway, the phosphate gets cleaved off of bisphosphoglycerate, given to ADP, and you get ATP. And then the other product is bisphosphoglycerate. 1,3-bisphosphoglycerate will just go to 3-phosphoglycerate. So it lost one of the phosphates. All right. So, and then it continues on in this pathway, and then you see another substrate level phosphorylation event further down here. All right. So then by the time you're done with stage 2, you get that pyruvate molecule, which is one of the key molecules in glycolysis. And it's what I think of as kind of the decision point. So it's at this point where there's pyruvate, where the bacteria has to, or the microbe has to say, okay, now I want to get the most bang for my buck. The best situation for me would to push pyruvate into the citric acid cycle next, if I know I have oxygen. That would be the way to go, because I'll push it into the citric acid cycle, and then I'll push it, the products of that feed into the electron transport chain. But the microbe might, do the assessing and go, oh, okay, I don't have any oxygen. So if that's the case, then pyruvate can get pushed into stage 3, which are more fermentation pathways. And again, the goal still is they will get some ATP. The artist didn't show it on here just because it would probably make the slide a little too busy. But if you push these reactions, there's lots of different options depending on the microbe. And when you look at it carefully, you can see that there can be some ATP production made through that. But this is a fermentation. When you talk about fermentation, what that means is that nowhere in this pathway do you see oxygen being used to accept electrons. Nothing like that's going on. There's no oxygen involved in this pathway. And then the other thing about fermentation is that you don't have complete breakdown of the starting molecule. So glucose, so to fully break down glucose, what we mean by that is you would have to take that sugar and break it down to CO2. That would be a full breakdown. And that's great. If you can fully break down a sugar to CO2, you get the maximum amount of energy from that. But you see here, you don't see CO2 showing up in any significant measure, especially as you work just down to pyruvate. And so again, fermentation is not terribly efficient. It's more efficient for a complete breakdown. All right, so that's another thing about fermentation is you just don't get as much ATP as compared to if you work the electron transport chain. Questions on this? If you haven't seen it in a while, do you have questions?
Yes, so we just need to know like what parts of this cycle right here. This is fermentation, so it's a little bit different than glycolysis. So like I'm not sure what part of this I need to know.
The stage 3, you mean, or all of it?
Just the whole picture right here.
So yeah, I certainly am not going to say, tell me all about glycolysis from start to finish, and you have to write this all out. No. Biochemistry classes, you might have to still do that. I remember having to do that in biochemistry. But for this, it's more of understanding, what's the big picture of stage 1? Well, you have to expend a little ATP to get into, that fructose 1,6 molecule. And then really what I want you to focus mostly on is stage 2 here, because stage 2 is where you get substrate level phosphorylation happening. And no, I'm not going to say which molecule is donating the phosphorus. I'm not going to even ask you that. However, you have to know this is a substrate and it's phosphorylated. So if you can just say, yeah, there are some substrates in glycolysis who will the phosphate gets cleaved off of them and given to ADP to get your ATP. So knowing that is important, but not the actual details for this class of which molecules and step by step. I'm not going to ask that. And then pyruvate is another key thing. You really need to appreciate that by the end of stage 2, you get this lovely pyruvate, and depending on what habitat the microbe is immersed in, they would then either shunt that pyruvate over into the citric acid cycle, and then after that the electron transport chain process will run. If there's oxygen, they'll do that. If there isn't oxygen, then they have to think about there's actually 2 different things they can do, and it's not shown on here. One of the things they can do is push more fermentation. So the green is just some other fermentation pathways they can keep running with. Another thing that's not on here is, and we'll learn about this next time, some bacteria can do what's called anaerobic respiration, where they don't use oxygen. They still have an electron transport chain, but oxygen isn't the molecule that they're working with in that process. And so pyruvate is kind of this decision point. You know, what should I do with it? Should I do aerobic respiration, anaerobic respiration, or should I just keep going with fermentation? So we'll talk about that, and we'll think about, you know, if you are a microbe and you're trying to maximize your payload of how much ATP you get, you would rank that. You will have a definite ranking of choices that you would pick from. So we'll get into that. I think we've ran out of time, haven't we? Yep, we're over time actually. So we'll stop here. So remember now, for tomorrow in lab, it's group 2 that's coming in. Group 1 does not need to zoom in or anything. You can do other things that you have to do for your life and your other classes or this class. Remember, don't forget about the field guide. Maybe over this long weekend, you could go out on some hikes and think about the field guide idea, and get some fresh air and hopefully some sunshine. All right. But tomorrow I will see group 2 in lab at 10 a.m. All right. Have a good rest of the day, everybody.
Hey, are you ending it right now? Could I ask you a quick question?
Go ahead. Yep, you can ask.
So like the field guide, that's for our like the independent research thing, right? We're trying
No.
to figure out, or no, that's different.
It's a separate assignment that's going to happen early on here, and it's when you do your field guide, I'm not asking you to take samples from their environment and assess, like come back to the lab with those samples. For the field guide assignment, it's more of, I want you to get into the published literature and rely on, you know, scientists who have already studied those types of habitats. What are they saying about those habitats? I want you to learn from the published literature what they're finding about, you know, that, you know, if you are looking at a stream or the lake or a forest system. Now, some students have asked, can I use my field site for my independent project later? That's fine. If you want to, you can. But for the actual field guide assignment, I will not ask everyone, yes, you have to take samples, blah, blah, blah. I'm not going to do that. It gets to be too much. All right. Does that answer your question, Kyle?
Yes, so I just find like a cool spot that I think is gonna have some like interesting microbes, and then just like keep that in mind for when the time comes.
Yes. And so,
Okay.
and you hit on when the interesting part, you want an environment that has some diversity in it. You know, maybe, and I like, it's usually a good thing to have, like, you know, some kind of forested system and some kind of water. Whether,
Yeah, that's what I was
you know,
thinking.
a stream or lake, or some people pick a forest system with a meadow, you know, open to the sun, you know, a meadow area, and then a lake or stream. And then some people deliberately go to like a polluted stream area and, you know, get into that kind of angle. So there's lots of options, but the diversity of the site is, I think, nice, because then you have more to say and it's more interesting for, you know, remember, you're trying to design a field guide for someone who's like a, you know, a geeky kind of citizen who loves biology and wants to take this field guide, go to your site and learn. And so you want to make it interesting and entertaining, but accurate, you know, accurate to what you're saying about the site.
Okay. And then let's see. There was one slide I remember from like way back. It was like the first lecture, and it had to do with like sunlight hitting hydrogen, and then it turns it into hydrogen gas. And I didn't ever like learn that process. And then I would try to go on YouTube, because I like to go on like, because I remember when way back when I learned like glycolysis and stuff, I watched these videos with this person, and they like pretty much stepped me through it. And I couldn't, is there like a video that I could watch of that so I can get it more in my head? Because I'm looking at it and I'm like, I'm not really sure what the heck is going on. So
yeah.
Like, what is it called exactly, so I can YouTube it?
Yeah, that's a good question. How would they name that one? Let me see if I can find anything. I might, you know, like sometimes the textbooks, they'll send you movie clips that come with the textbook. So maybe in some of my microbiology texts, I can find a video that they've, I can't promise anything, but I will look and see if I can, or if I can't do that, maybe, you know, find a search term that you would be able to find something on YouTube. And then certainly we can talk about it, you know, either in lecture or lab or during office hours, we can work through it too. So I know a lot of students, you know, it's their first time seeing that reaction, and it, you know, if you don't have someone explaining it to you, it's hard to figure it out.
Right.
Yeah.
And
So,
then as far as the process of the hydrothermal vents, do I have to know that mechanism too, or is it just the concept of it, of like how it's alkaline water sort of, because there's a lot going on in that image, and I'm like,
not even alkaline water. I'm not even going to ask you that. It's more of that you could describe that they, you know, the leading hypothesis is that they think that's where life originated, was deep hydrothermal vents or the ocean floor, because it had, you know, if you can understand, there's high heat in that area coming from that superheated water. And then there's the right mixture of minerals and surfaces. That's really
Yeah.
that's about it. I don't want you to really get into, you don't have to tell me what kinds of ions and minerals and all of that. You know, it's just, there was the right mixture of surfaces, heat, and, you know, the right nutrients and minerals available to allow for these protocells to develop.
All right, sweet. All right, I guess I'll try to figure it out myself, and if I get the process in my head, I'll like try explaining it to you in lab on Friday. And
Yes.
if you tell me that I'm right or wrong.
Yes. And that's an excellent way. What you just told me is you're going to self-test yourself. You know, you're going to study it for a little while, and then you're going to self-test yourself. And students really need to do that before they walk in on exam day, or, you know, before they take the exam, because if you haven't figured out what you know and what you don't know, you know, it's like you have to do this pre-testing before you walk in on and take the exam, so that you are aware of, yeah, I got that down, but I'm shaky on this. And so certainly, yeah, Friday we can talk more about it, and I'll give you some feedback.
All right. I guess, and then one more thing is, so when pyruvate is at the end, we have pyruvate, and then so when fermentation starts, it like shoves it. It doesn't shove it into the electron transport chain, does it? It doesn't. What is it? What is it? What
No,
exactly does it do?
pyruvate, the next step for pyruvate, if there's oxygen available, what happens is pyruvate will go into the citric acid cycle.
Right, but
And then some of the molecules that get made in the citric acid cycle, those get shunted over to the electron transport chain and fed into the electron transport chain.
Okay.
So I haven't covered that yet. We just, you know, got through glycolysis today. So the very next time we meet for lecture, I'll be talking about this idea of what happens to pyruvate, and I'll work you through that. Okay.
Sweet. Okay. Well, I'll see you on Friday.
Okay. Sounds
Tomorrow,
good.
I guess. Yeah. All right.
See you, everybody.
Thank you.
Mm-hmm.